Today I want to start with just tension in general, what is meant by that. What you got to realize is when your muscle is relaxed and it is sitting there, it is being, these sarcomeric units, these sarcomeres that are in your muscle fibers, they have what's called an optimal length in terms of, how far, if I can imagine that I'm holding a sarcomere. So here's a Z line, here's a Z line. There's actin anchored to both of these Z lines, and there's myosin sort of down the middle in between them. When that sarcomere contracts, it comes together, right? And the little myosin cross-spread, just grab onto that actin and ratchet it towards the center, and you get a contraction. What you got to realize is if you can take that sarcomere and pull it apart far enough to where the myosin, there is no overlap now between the myosin and the actin, obviously you're not going to get a contraction, right? What you've done is overextended it past the point where cross-bridge interaction can occur. Now you can also see that you're going to witness the biggest degree of contraction whenever you have a sarcomere that's spread out as far as it can and still be able to have cross-bridge interaction. Okay? If you can take that and spread it out as far as you can and just have a little bit of myosin here where it can just grab onto that actin and start that process, when that contraction starts, you're going to see a much bigger contraction. Right? Does that make sense? Okay, so what we have is a resting state called, I'm going to call optimal length. Optimal length is how far apart these guys can be and still result in a nice big contraction. Okay, what length is there that will result in the most optimum contraction? And that is called the optimal length. I'm going to bring this back a little bit at the end of the lecture when I start talking about smooth muscle, but I'm talking still with skeletal muscle here. Okay, so our optimal length is defined as whatever length is there that will give you the maximum opportunity for cross-bridge interaction. Okay, if you get them too close, now realize if the myosin here is in between, and my fingers are going to be actin, if they're really close and there's a whole bunch of myosin in here grabbing onto a whole bunch of actin, when it contracts, it's not going to go very far. Right? It'll still be a good contraction, but it won't be evidential. You won't be able to see it as well. It won't be a maximum contraction. Okay. Now, obviously, also that's what's going to control tension. Is so it says at lengths other than the optimal length, not all cross bridges are able to interact. Okay. So. Some other thing that's going to control tension is just general fatigue and how thick the fiber is. In certain muscles, the fibers are thicker than others. And you can certainly thicken some of those fibers yourselves with enough weight training and that kind of thing. I'll talk about that in a little bit. But the thicker the fiber, the more tension that can be generated by that fiber. You've got more actual proteins. Remember when we take a muscle fiber and we cross-section it and look inside, there's a three-dimensional array of actin and myosin all around in there. And the more of those proteins your body builds, and the more you assimilate into one of those fibers, the more chance you have for a greater tension of those fibers. Okay, that's the strength, so to speak. Now, this tension obviously can be transmitted to bone. Everything's attached. So whenever you have a muscle contraction, realize that the tendons holding those muscles to the bone are also they're not contractile units. The tendons aren't contractile, but they are attached to the same fibers that have contractile units in them. So when they start to contract, that tension gets pulled, and those tendons are anchored on to the insertion and to the origin, right? And when that does, that tension is created and it passes on into the bone. So you can actually create the muscle tension can be passed on and absorbed by some of the bone as well. So there is tension there. All right, so you should know this from basic anatomy: origin, insertion in terms of muscles. The origin is going to be where the muscle is anchored to a non-movable type of position versus down here, the insertion, like if you're looking for origin of triceps and biceps, the insertion is going to be attached to the movable end. Okay, so this is obvious. I shouldn't even have to say this, but if the muscle tension can overcome a load, obviously it's going to pull the insertion towards the origin. Okay. Now realize you can create tension without actually shortening that muscle. I'm going to talk about that here in a second too, like called isometric contractions. You guys, anybody in weight training or anything like that would know the difference between isometric, isotonic. It's the difference between like pushing against a wall for 30 seconds as opposed to doing reps and curls and that kind of stuff. One's isometric, which is sort of a constant pushing. One's isotonic. I'll show you that in a second as well. And here it is. So in an isometric contraction, the muscle tension developed is less than its opposing load. Obviously, if you go and push against this wall, you're not going to be able to push it down. Okay? So whatever tension you develop in your muscles as a result of pushing against that wall is obviously going to be less than the opposing load, the opposing force that's coming back at you. If that's the case, we just call it an isometric situation. as a result, what you will see is the muscle will not shorten. Isometric means equal length. It's going to stay the same length that it was, okay, because you're not allowing the muscle to shorten. And thus, it's not going to lift any objects as a result of that. Now, with isotonic contraction, you can develop tension greater than the opposing load. The muscle will shorten as a result of that, and when it does, it moves. It creates movement, and it can, if that tension is greater than the opposing load, you can move that. Okay. All right. Now, the velocity. We've talked about like twitches, fiber twitches, and that kind of thing. Certain fibers have the ability to, when I say a twitch, I'm not talking about just a flex or a contraction. I'm referring to the individual fibers. A muscle twitch is when one of those sarcomeres come together. Okay? Now, collectively, if you have a whole bunch of those things happening, you can create what's called a contraction in the entire muscle. Now, as far as the velocity of, we've got different fibers that can twitch faster than others, and all of this plays into how fast a muscle can actually contract in a given circumstance. This should be a no-brainer as well, but it says, to put it in fancy words, the velocity of muscle shortening is inversely proportional to the magnitude of the load. Whoa, what's that mean? When one goes up, the other goes down. The heavier the load, the longer it takes for the muscle to shorten, right? The lighter the load, the faster the muscle shortening. So inversely proportional means when one goes up, the other one goes down by the same factor. Like you can see on this graph. As the load increases, you can see how the velocity of the shortening constantly decreases as the load increases. Okay? That's, again, should be common sense. Okay, concept of work. Now, you all have your own definition of what work is and a working definition that you apply to your life, but from a scientific perspective, work actually has a definition. Work is calculated by multiplying the magnitude of the load, in other words, how much force is associated with something, times the distance over which that load is moved. Work is as I stand here, it's sort of a relative term, because this is where it's different than what you would. As I hold this and I ask you if I'm doing work, your answer may be yes, because you know that it takes ATP, it takes energy for me to stand here and contract the muscles that are contracting to hold this thing up. But according to science, there's really no work being done in this particular, at a big level, little levels maybe, but at a big level, there's no work being done because I'm not applying a force over a given distance. Okay, there is no change in distance. Technically, here's what's weird too. In terms of science, okay, whatever my force is, that's weight. Okay, if I'm moving my weight from here to here, I've moved it over a given distance. I've performed work to do that. However, if I walk right back to where I was at the exact same spot, overall I've done no work. That's crazy. It doesn't make any sense to you. But mathematically, force has to be carried out over a given distance. And when you get into physics and that kind of stuff, distance is displacement. How far away are you from the given point where you started? Okay, so if I go from there to there, I've got a measurable distance from where I started versus where I ended. Oh, if I go from there to there and back again, my total displacement is 0. I ended up in the same spot that I started. Anyways, it's just a mess with your head. Don't worry about it. Anyways, just realize that if you take a force and apply it over a given distance, you've performed work. Okay, so force times distance. W equals FD. To that end, much of the energy that we apply in our body to perform work is actually lost in the form of heat. We have a constant internal body temperature, right? And there's a lot of things that go into maintaining that. We only get of all the energy that we spend on trying to do work and move forces over a given distance, only about 25%, only about a quarter of all of that work is actually realized and is used to do that. And in the process of creating that, about 75% of that energy that is headed towards doing work doesn't end up doing work at all. It actually just gets converted into a waste heat energy. You can think of it as a waste, but realize we do have to have some heat energy to maintain our body. So it's pretty inefficient. It's only about 25% efficient in terms of getting out the work out of the energy that we expend to do that work. All right, now there's a lot on there. I'm going to use this picture to explain it. Okay. Now stay with me on this one. Looks pretty fancy, but you already know all of this. You just don't maybe recognize it already. All right, now here's our bloodstream. What this is going to be is what happens inside of a muscle fiber. Okay, here's the bloodstream. Notice what can travel through that. We know oxygen, carbon dioxide is traveling in the bloodstream. We know that glucose is traveling through the bloodstream. So let's start somewhere. Let's start with glucose. As glucose travels through our blood and goes out into many places, including our skeletal muscles, it can diffuse in. We've talked about how all this can happen. It's not just regular diffusion. Glucose happens with facilitated diffusion. Insulin's got to be present. Some other cellular proteins have to be there to control whether or not glucose can come into a cell. So assuming that all that is skipped and glucose gets into the cell. Okay, first of all, where did the glucose come from? Number one, digestion. We're going to talk about the digestive system later. Everything is converted down into simple chemicals like glucose. Where else can it come from? Well, you don't just eat something. Say you eat a big bowl of spaghetti that has a lot of carbohydrates in it. Ultimately, you get all converted into glucose. Okay, well, a lot of it gets converted into glucose. As you load up on all this glucose, you got to realize that it doesn't just stay in your bloodstream. Okay, it doesn't just float around until it's all used up. Your body realizes how much of it is there and controls how much is in the bloodstream at any given time. This is the premise behind the whole diabetes thing, right? You know that sometimes the blood sugar can get high, sometimes it can get low. Your body, in a normal circumstance, in a healthy individual, will maintain a certain window of blood glucose levels. Now, that may mean that your body has to store away some of the extra glucose or pull out some of the stored glucose. Okay? To store it away and hide it away, our body converts it to a form called glycogen, which is like, I use the analogy of the toilet paper in the past, didn't I? Well, glycogen's like the toilet paper, and glucose would be like the little squares. So glycogen gets assimilated, glucoses get kind of stuck together and stored in the liver. And then as the blood circulates through the liver, the body recognizes, okay, glucose levels are dropping a little bit. I'm going to take some of this glycogen, pull it out, convert it, and release some free glucose into the blood. That's what happens between your meals. Okay? At the same time, if there's too much, it'll say, I need to pull some out. I'm going to convert it into glycogen and store it away. Now, here's what's interesting. There's two places in your body, really, of any significance where this occurs. Number one, in the liver. Number two, in your muscle cells. Your muscle cells have the ability to store glycogen in them. And think about it, and it makes sense as to why that would be the case. You use your muscle cells, they use a lot of energy in your body, right? They burn a lot of ATP. They have to use ATP for all the contractions that you do all the time. So it makes sense then that your cells have developed this specialization to be able to hold glycogen in them for immediate use if that glucose becomes necessary. Okay? So that's where this muscle glycogen comes from. Some liver glycogen could contribute to some of this blood glucose. It just depends. So in any case, we've got glucose coming in. All right, so what happens when glucose enters a cell? In this case, a muscle cell. Same thing as it does with all the cells. It starts to go into the whole turn it into ATP thing. Go back to like the second chapter or whatever. Glycolysis. We know that glycolysis occurs because you got to realize a muscle fiber is a muscle cell. So in it are all the organelles. Okay? In between all those sarcomeric units that we've been talking about, realize all the organelles are there. I just haven't showed them to you on those diagrams. The mitochondria are there. Muscle cells are loaded with mitochondria for obvious reasons. All the other stuff is there. So in the cytoplasm, the same enzymes that were responsible for in the other cells can cause glycolysis to occur. So that 6-carbon sugar gets split, right? And you get pyruvic acid from that. Here it is. Now, when pyruvic acid gets split, we said that for every one of those, there was 2 ATP that was generated. So some ATP we're going to just sort of classify that separately. We're going to pull it out and say it's headed over this way. We can use it now to keep track of it. Now, what happens if glycolysis continually occurs? You're producing a lot of pyruvic acid, and there's no oxygen. Remember, everything gets backed up when there's no oxygen. So pyruvic acid starts to get converted into lactic acid. Okay? So if there's no oxygen, lactic acid can start to build up, and then what happens is it doesn't stay in that cell. Okay? It gets secreted back into the bloodstream, and as such, probably makes its way very quickly to the body's muscles, especially in that local area as it gets in the bloodstream. It starts to build up, and realize what happens, okay, why lactic acid contributes so much to muscle fatigue and soreness and that kind of stuff is because acid has the ability to take the structure of proteins. Proteins are a bunch of amino acids, right, all stuck together. And what proteins, finalized proteins are, they've been snipped and pruned They've been squashed together, twisted, crammed up, and wadded up, and everywhere they're close to each other to hold that wad together are little tiny bonds, little disulfide bonds that keep this protein intact. In an acidic condition, when acid levels start to rise, the hydrogen ions from acids can start to interact with those proteins and pull them apart and fray them out a little bit. And when you do, that's called denaturing that protein. The protein will not work anymore. Okay, so if these are proteins that are involved in second messenger systems, are they actin and myosin themselves? If these are all types of proteins like that, they will be affected if lactic acid builds up, and muscle contractions won't work as well. Okay, they will not contract like they should. They will not pull calcium back out of the way, back into those sarcoplasmic reticulum to relax the muscle like they should. So that it gets fatigued very quickly, if that's the case, and lactic acid starts building up. Okay, so if, however, oxygen is present, we know that pyruvic acid can enter into the Krebs cycle, right? And then from the Krebs cycle, that gets converted. Acetyl-CoA goes down to that little Ferris wheel-looking thing. It all spins around, and ultimately what gets kicked out are hydrogens, right? And the hydrogens get carried over to the membrane in the mitochondria, up at the edge of the mitochondria, and they enter in between that membrane space, and then all of a sudden you get this shuttling. The electron gets stripped from those hydrogens. They get shuttled along. They come shooting through those chutes, spin around that little propeller, and form ATP. Okay, a bunch of ATP when that happens. All right, that's called oxidative phosphorylation. You remember, because phosphorylation is adding phosphate to something. So whenever those hydrogens were shot through that little tunnel on that membrane of the electron transport chain, what they actually did was took a phosphate group and positioned it next to an ADP to make an ATP. So that was phosphorylation. Well, oxygen had to be present because why? What does oxygen do? Do you remember? Why is oxygen so important in that process? That's right. It is the final acceptor on the electron transport chain. Okay? Because when hydrogen got in there, it got its electron stripped, and then that electron got passed from one situation to the next to use it for energy. Okay? All right. So all of that being said, this is all happening in the muscle cell as well. So with all of that, you're cranking out a bunch of ATP. So we got some ATP from this, some ATP from glycolysis. We're headed over that way to use our ATP by the muscle. Okay? Now I want to point out something here. Notice other inputs that can go into oxidative phosphorylation. I say protein. I will also say fatty acids. Okay? Our body has the ability to use and digest proteins and fats as an energy source. Now, it's not designed to be our primary means. Okay? Fat supplies are for reserve purposes. That's the purpose of them, for having in our body. But realize that when I want to point out something. Whenever our body breaks down fat stores because glucose isn't enough, or some other reason in that person's body causes it. In some people, there may be more of a reason, a more enzymatic metabolism thing happening to where they break fat stores down more easily. Who knows? For whatever reasons, when these fat stores get broke down, they get released into the blood first, because these are from other cells, adipose cells that are outside of the actual muscle fiber. It itself is not storing this fat storage. Okay, so what happens is they have to release those contents, and they release them in little chunks of fatty acids. Okay, if you think back, I don't know if you remember or not, I talked about phospholipids in the phospholipid bilayer of the cell, and I said that there was like a head and it had two tails coming off of it, right? But an actual these tails coming off of it were actually fatty acid chains. Okay. Well, anyways, these little fatty acids can be released and travel out here into the blood. That's a problem. We have now an acidic condition now. The more fat that is broken down, the harder it is on your body to maintain homeostasis, because you're approaching a situation. Acidosis could be a risk factor at this point. The more that you do this. At first, your body deals with it. But if you do it consistently over a long-term basis and you rely on solely fat storage breakdown for that purpose, realize the homeostatic challenge you are posing to your body when you do this. So this could affect proteins at many levels. Okay, so some of these fatty acids could be brought in and actually go through that whole electron transport stuff that we talked about. I'm not going through the mechanism by which that occurs. Just realize they can do that. And when you do, you get the same products. can actually use, just like glucose would give you that product, fatty acids could too, and yield the same thing down here. Okay, water and carbon dioxide are the end products. Also, protein. If you, especially if, now, it would be nice if there was a nice sequence that our body followed. If it was like, okay, no carbs, I'm going to get rid of all the fat. Okay, out of fat, I'm going to get rid of the protein. Well, that would be nice, maybe, for people who go on diets or whatever. But realize it sort of all fades together. If there's a shortage of glucose, the fats probably, more fat will get broken down, but don't kid yourself, protein can get broken down as well. So when protein starts to break down, look what it breaks into. Amino acids, right? So as long as there is a release of any form of acid, you're back in the same situation I talked about over here. Certainly it's a viable form. It'll produce this energy. But I mention this especially too because with some of these diets that are out there and the fads and those kind of things. Think about Atkins diet. One of the reasons that it is dangerous is, at least over long term, it depends on, you got to weigh what it is, where you're at now versus where you're going, and is that a risk factor that people are willing to take? Consult a physician, that kind of thing. So I'm not saying it's horrible or bad. I'm just saying keep in mind some things when you're doing this. The more you know about what you're actually doing, and the more that you know about your body and what's happening to it, the better off you're going to be, right? With an Atkins diet where you basically eliminate carbohydrates as much as possible from the diet, you rely upon these methods to get your energy, fatty acids, and even protein. And as a result, if that was carried out over long term, you're going to have a couple things, it's going to be in response to what's going on. Number one, elevated acid, general pH of the body. Now, your body has buffer systems, and it'll act against this. And for a while, you can go on that for a while, and probably nothing will ever happen in terms of severe consequences. But say you stay on this long term, it was not intended to be a long-term diet. And anyone says it is, that's kind of foolish. But it can't be a long-term diet. Your body's not designed that way to handle it as a long-term situation. That's too dangerous. You're playing with fire here when you do that. Now, I was going to say something else. Oh, another thing that comes about with the breakdown, especially of these guys right here, is you get ketones released. Ketones are precursors to organic acids. I don't expect you to know the chemistry behind that. That's fine. But sometimes there's a lot of, it's not this simple, right? It's not black and white. There's a lot of in-between things that can pop up and occur, and intermediary products and all kinds of crap like that. But when proteins, especially proteins, even these guys, but especially proteins get converted into amino acids, little molecules called ketones. They're a group of chemicals, so there's a bunch of different types of ketones can occur. Now, ketones, like I said, can lead to acidosis because ketones are precursors to things that could easily form acids in the body as they're floating around in the blood. And think about, for those of you who are familiar with this, one of the things that you do to test for, like diabetes, for example, right, is a ketone test. If ketones show up in the urine, it's a good indication that there's a cause, a red flag to be a concern if this person has diabetes or not. You tell me why. Yeah, the ketones are obviously spilling over into the urine. That's why they're showing up. But my question is, why is there an elevated level of ketones in the body? No, stay with it. Just keep going. Your body. You say your body. That's one solution. It could be that your body's not producing insulin. If that were the case, explain to me through this whole process that would lead to the production of ketones. Well, they can. Let's stick with just from the glucose side of things. If in fact the reason that ketones are spilling into the urine is because of some sort of glucose problem, explain that to me as to why that would be the case. So basically, the basis for the test that you're using. If you're going to give this test, why are you giving it? Why is it useful? How does it work? Not necessarily. I don't care how the chemical interaction occurs. But, huh? Too much what? What? You're just throwing out fancy words, right? Now think about it. Maybe you're not understanding what I'm going for. If you have too much ketones in the urine, the reason is because your body is doing what? maybe. I mean, we don't know that yet. All we know for sure is what? That's right. It's breaking down protein excessively. Right? So if your body is breaking down protein excessively, what could that indicate? Based upon what I've just said. If you're breaking down protein excessively, okay, not enough carbs are getting into the cell. Right? So if you don't have enough glucose, carbohydrates getting into the cell, your body's going to say, something's going on. I've got to do something different. Now, your body doesn't realize everything that's going on in terms of, it may not realize that insulin is the problem. It may not realize that the receptors that interact with insulin, the insulin's fine, and it's just the cellular receptors that are the problem. It doesn't know that. All it knows is how to respond homeostatically. And what it is going to do is respond in a manner that will try to rectify this situation. And in so doing, it will start breaking down proteins, thinking that it needs a reserve energy source. And when it starts breaking down proteins, you're going to get an elevated level of ketones in the bloodstream. So that's why it's indicative of maybe a diabetic type of situation. Okay? Something is wrong with the body's processing of glucose. Now, you have to do other things to determine whether the insulin itself is okay, whether it's faulty, whether it's like a type 1, type 2 situation, and all that. And we'll get into that when I talk about digestion. Yes and no. I'll explain that when I get to the urinary system. It has to do with osmosis. And it has to do with when you load up solutes and you change concentrations, water follows accordingly. And so what they end up doing is increasing the solute particles in their urine and drawing in more water from the body. So it causes the bladder to fill. It causes more urine to be produced. And as a result, you get more copious urination. That kind of thing. So, not only that, but it smells fruity sometimes too. Ketones are, if you have like acetone and ketones, these are related chemicals, smells like, no, it does, really. I mean, in a really, really bad patient, and like I said, I don't have any clinical experience, but some things, I mean, I've got to experience, I suppose. If you have a really, really bad diabetic person who's being treated, in bad situations, you could actually almost to the point where it smells like fruit punch sometimes. It could be really bad. If you've not had that experience, talk to someone in your field who works with those type of patients, and they could probably testify to that.
If you have like what?
If you have a high level of ketones in the urine, yeah. Not just because you're so wonderful that your pee smells like fruit. I'm not saying that.
No, I have type 2.
Okay.
You're saying this, and they're all looking at me like, oh, your pee smells.
Well, hold off on that. Let's wait until we get to digestion and we talk about the differences between type 1 and type 2, and some of those questions will sort of answer themselves as we get along, as we go farther along.
Isn't having the ketones and the acidosis, isn't that a way that they tell you can tell if you're
Well, sure, because the whole point is that your body is breaking down proteins. So if you're breaking down proteins, the ketones will be indicative of that. So that means the diet is accomplishing, at least at a metabolism level, what it's supposed to be doing. So I don't know. Okay, I'm getting bogged down. The good questions, though. Good questions. But when we get into digestion, we'll open up a door that we can talk about more about this. Okay. All right, now, one other thing. I don't know if anybody has sons, daughters, especially at the middle school to high school level or whatever, when they're starting to get concerned with weightlifting and that kind of stuff. I used to coach baseball, and I had some of the guys that were big into lifting, and we had a lifting program and everything like that. But one of the times, I always stretched them if they were going to take any supplementation or anything like that. Make sure you talk to me about it. Make sure you know what you're doing. First of all, make sure it's legal. Second of all, don't just take things because they're on the shelf and you can take them. You got to understand what you're doing, because, like, for example, creatine. We've probably heard of that, right? Creatine powder, supplementation, all that kind of stuff. Creatine phosphate can be taken as a supplement, but you got to realize that if you take it, what it does, and the only way you're going to get anything from it. Okay? You can't just take it and wake up and expect to just start getting bigger just by going through the day. It's not going to work like that. Because we've talked about this ATP, right, and where it comes from. So we're going to be dealing with that ATP source. Another source of ATP is that we have inherently within our muscle cells, creatine phosphate is present. Okay, creatine monophosphate. And creatine monophosphate, what it does is takes ADP that's been used from ATP breakdown and recycles it, and it donates, look at what it says here, creatine phosphate. It'll donate this phosphate group to ADP to make ATP. Okay, so that's what it does. And when it does that, it splits off, and creatine then can be either, usually at that point it could be recycled, but usually it can actually appear in the urine and actually be excreted and that kind of thing. So it just depends on the product where it goes. And that's not what our concern is. What I want to point out is if you take creatine monophosphate, for example, as a supplement, what advantage is there? How is your body going to deal with that from a physiological perspective? Realize that all you're doing is creating a means by which you can have more ATP in your muscles. That's it. Okay, so if you take creatine and you don't work out or you don't hit it extra hard, okay, the whole point is so that you can intensify your workout, so you can have more energy associated with the 20, 30 minutes that you're going to put in, or whatever, 45 minutes, whatever kind of conditioning you're going to do. If you do not utilize that, it's like putting in stuff that the only way you're going to get anything out of this creatine is to burn up all the ATP that it uses. It doesn't allow you to build bigger fibers directly. It doesn't give you the stuff. It's not like steroids that are going to build protein, things like that. It's going to give you more energy to work out, basically. It's going to give your muscle fibers more time. They're going to fatigue less easily is what's going to happen, but not if you don't work out. Okay, I got guys that I've had kids take it before, and they think that I have one guy who used it well. Other people that would take it and they think they're going to come in there and just do just push through their reps or whatever, do what they're supposed to, and go home. It's not helping. That doesn't help at all. Okay, if you're going to take it, you got to go in, you got to hit it hard, and you got to utilize that extra energy and that extra ATP that your body's doing. Otherwise, it's not even beneficial. I think it depends on. I don't know exactly how all that works. My brother was into all that, but he worked at the GNC. You take it. You can take it like in the form of like shakes and that kind of stuff too, and mixed in with powdered drinks. You take it in the morning. You can take it before you work out. I don't know all the regimens that exist and the possibilities you had to choose from. I don't know. Yeah. Diet drugs, as in. They have their creatine, or I don't know what the chemical is. I guess. Some, to be honest, some like weight loss pills, and again, this is not an area that I know a whole lot about, but some of them is just actually like derivatives of speed type of drugs, like methamphetamine types of drugs that are not nearly as potent, but certainly could be. If abused. But mostly what that will do is just speed up the body's metabolism in a large-scale sense. Possibly damaging in terms of homeostasis, but that just depends. I'd have to look into it. I don't know a direct answer to that. Okay, now out of all of this, here's our ATP that's generated. Where does it go? Bingo. To contract. Realize how fast this is all happening, folks. This is happening in like fractions of a second, everything that you see up here. So we take all this for granted. Myosin ATPase activity is happening. Cross-bridge interaction is what this ATP is going to be used for. Okay. Now, to get a relaxation from that, remember all that calcium that was on there that had to pull those binding sites, that troponin tropomyosin complex, away from the actin. The only way that's going to close and get a relaxation is to suck away all that calcium back out and store it back up into that sarcoplasmic reticulum in the muscle. So that's going to be your relaxation. Then the ADP is released and then could be reused again, deteriorated, who knows. There's a lot of things that could happen. So a lot of stuff going on. Now you know all about it. You can tell everybody about it. Okay. Oh, come on, you followed that, didn't you? You could. Well, that's the point. I mean, you can't. The purpose of a lecture is not so that I can sit down individually and make sure that it all makes sense to every single person. The best I can do is come up here and give you an example of how at least it makes sense while you're following me. And then it's up to you at that point to either talk with me or someone else or put in the time outside of class to to try to make sense of it for yourself. So, all right, and we talked about muscle fatigue. Muscle fatigue is when basically your muscle doesn't respond to the same degree now as it once was doing. Okay, you stimulate it in the same way. I don't know if you guys have ever done like a conditioning weightlifting type of program, but one thing that you'll find out, is it gets frustrating after a while. You start off with your reps or whatever, and then you're working, and all of a sudden it doesn't make any sense. You're putting in just as much effort and getting far less in terms of what you're getting out of it. Your muscles become fatigued. Now, what would cause this at a chemical level? Okay, there's a few things. I've highlighted them up here. A local increase in inorganic phosphate. Somehow, I don't know. I don't know the exact. I don't even know if your book talks about it. The exact chemical process by which this occurs. But what has been shown is from a whole lot of ATP breakdown, as your body is using that to make this contraction happen. Inorganic phosphate. In other words, one of the P's off ATP, a single P starts to build up, and a lot of them start to build up faster than breaking down faster than they can be regenerated. When that happens, your calcium doesn't function properly. It doesn't get where it needs to be. Your cross-bridge interaction doesn't function properly. So no matter how much nerve input you're putting, how much you're trying, you're not getting that degree of contractility that you once were getting because of this buildup. Lactic acid builds up. Now remember, I said what happens. The biggest thing about acids and how they affect biological systems is the proteins that are involved in biological systems. Now we have special proteins in our body that whenever they are involved in a chemical reaction, but they aren't used up in the chemical reaction, they're just there to help things happen. We call those proteins enzymes. Okay? Enzymes are proteins, and as such, lactic acid can screw with enzymes. Okay? It makes them not work as well. And obviously, we've got way too many cellular processes that happen that depend upon enzymes to work, so you can see the problem here. All right. Extracellular potassium can play a role in this. Realize that for a muscle contraction to occur, there's got to be a nerve input, so to speak. It doesn't allow it to contract like it should. Glycogen energy reserves. You've got some stored glycogen, I said, inside these muscle fibers. If you use it all up faster than it can be replaced, that's obviously going to contribute to muscle fatigue. It's not going to have energy available for the muscle contractions that you need. So what happens is we create what's called an oxygen debt. And that means that really the only way to fix the problem is to stop, rest, bring in a whole lot more oxygen than what you're actually using at that particular point. And then it takes us back to this screen so that all of this doesn't get built up. And instead, we can pump in more O2 and take some of that lactic acid, convert it back into pyruvic acid, and hit back on this side of things again and replenish all of this kind of stuff. Okay. There is something called central fatigue, and that has nothing to do with the actual muscles themselves, but rather the nervous system that feeds those muscles. If the nervous system is not functioning properly, you can obviously imagine and see how the muscles will not contract as either. Okay, so that could certainly be one cause of it. Usually it's psychological, and usually it's biochemical in nature. Something's wrong with the movement of the chemicals involved to make this nerve transmission happen. Okay, here's the three big groups of muscle fibers that exist, how we classify them. Of all the muscles that are out there, in terms of skeletal muscles, we've got what's called slow and fast oxidative and fast glycolytic. Guess how they get their name? They get their name by how they get their energy. Oxidative fibers, the difference between slow and fast is obvious. One contracts much faster than the other. Oxidative means they rely on oxygen, and they use oxidative phosphorylation, the Krebs cycle, all that stuff to generate most of their ATP, and that's how they function. Now, you do have what's called fast glycolytic fibers. These are fast-moving, and they actually get a lot of their energy and can work very well under anaerobic type conditions. Conditions where glycolysis is the primary means of generating the ATP for the muscle contraction. Okay? Now, generally we have all of these. Oxidative fibers, it says, are red with a high concentration of myoglobin. Myoglobin is sort of like hemoglobin. Myoglobin is special. Myo is in response to muscles, right? Within muscle cells, there's a special carrier that actually allows oxygen to be transported more efficiently inside muscle cells because it's so important to do so. Okay, so that myoglobin does not float around in the bloodstream. It's local. It stays inside the muscle cells. Hemoglobin, of course, stays in the blood and travels all around. So. Okay, now this brings us to an interesting point here because I think of a guy I used to work with when I worked at Pioneer. I won't say his name because I don't know who will ever be taking this class. But he was a very large individual, not so much this way, but this way. And you think about physical stature, you think about strength, and you think about when someone looks can be intimidating, right? You think about how big and buff someone is, you think they're really strong and that kind of thing. But it's funny because sometimes those guys, if they don't train properly, have no endurance whatsoever. None. I mean, they might be able to grab your throat and squash it, but you take 3 steps, they could fall over, because they have no endurance. And the reason for that, and certainly cardio and endurance should be part of the training, right? But you got to realize is that the reason for that is because the types of fibers that are used in different types of training. Let me show you what I mean. Regular endurance exercises like jogging, cardio, aerobic stuff, okay, promotes improved oxidative capacity in oxidative fibers. The fibers that are already oxidative, whether they be fast or slow oxidative fibers, rely on oxygen to work. So what you do when you condition is you take those muscles, whether it's the muscles that are involved up here, your heart muscle, it's all muscle, okay? You take your muscles and allow them more practice and get them better at more efficiently using the oxygen that is available. So that even under lower oxygen situations, they're really good at using oxygen, okay? So you're going to improve that capacity. They're going to be able to store more oxygen. They're going to be able to more efficiently use the oxygen that they have. And that's what building endurance is, okay? I used to be in shape. Some of us used to be. Maybe some of you still are. I don't know. But I know for me, that's no longer the case. Okay? I used to run miles and miles and miles at a time. Now the thought of that makes me just tired. I don't even want to run anymore. If I can't drive my truck to get there, I'm not going. All right, or my four-wheeler. But anyways, now, if you do high-intensity resistance, if you go in there and you work out 5 hours a day, but all you do is bench and shoulders and whatever, any of these things, as long as that's all you do, then you're going to get beach muscles maybe, but what you're not going to get is endurance. High-intensity resistance is anaerobic in nature because what it targets, and the reason that this is what it does is because you can see the results of this. When muscles get bigger, you got to realize what you are not doing is growing more muscle fibers. When you increase your muscle mass, you are not adding more muscle fibers. What you are doing is taking the fibers that you have and thickening them. Okay? You're adding more proteins to the inside of those muscle fibers to get more sarcomeric units is what you are doing. Now, the ones that are going to widen and get thicker in diameter are going to be those glycolytic fibers, the ones that are anaerobic in nature already, okay? Not the oxidative fibers. They generally don't get bigger and bigger. The bulk that you see with muscle mass and strength has to do more with glycolytic fibers than it does oxidative fibers. However, the endurance has everything to do with the oxidative fibers. Okay, so you can see that there's a dichotomy there, and there's a relationship that has to be exercised independently. So what you get is, like with weightlifting, promotes hypertrophy, excessive growth of fast glycolytic fibers. That's why it's intense. That's why it's fast. It's a whole different type of workout. These fibers increase in diameter. You get no new fibers. You just stick in the ones that you have. Now, testosterone. Testosterone has the ability, and this is where anabolic steroids come in. Realize that anabolic steroids are the same chemical precursor as testosterone. Okay, I'll talk about when I get into hormones. I'll explain that a little bit more. But testosterone promotes protein synthesis for this increase. This gives you more proteins to make more filaments inside those fibers to get them bigger. However, the fibers that you are growing are not oxidative fibers. They are glycolytic fibers, and as such, you are not helping your endurance. Okay. Let's see. Like I said, there's fast twitch and slow twitch fibers. That just depends on whatever nerve is innervating them to tell them, what type of message they are receiving, and thus how fast they're going to twitch. We all know that muscles atrophy when not used. Some of us can speak from experience on that, I suppose. There's disuse atrophy and denervation atrophy. Disuse means obviously you're not using it, so the actual the problem means that you rest with the muscle itself. I mean, it wasn't being used, it wasn't being exercised. As a result, it loses its contractility over a period of time. However, realize that atrophy could be because not just because you're not using it, but because the nerve supply feeding it doesn't properly relay the impulse for contraction to occur, in which case that could be denervation atrophy. And skeletal muscles do have a capacity for limited repair. They do have the ability to repair themselves to a certain degree. Okay, bottom line here, I'm going to try to speed up a little. Bottom line here, you don't have to know all of this, but as far as the neurons that are controlling the different types of muscles, so that's why I say motor neuron output, the feed that comes out to control these muscles that are being contracted. There's basically three major inputs by which that occurs. Okay, you've got the spinal reflex pathways. That's easy. That's non-cognitive. You don't really think about it. It just happens. There's an afferent impulse comes in, gives a U-turn in the spinal cord, goes right back out to the effector muscle and causes a contraction. Okay, that's one input. Another one is what's called the primary motor cortex. This is more cerebral. This is more thought. This is more complicated. Especially, like I talk about how it has more to do with the intricate movements of the hands, the little things, why you can sew, why you can take a cross-stitching thing and stick a needle into a specific square and pull it out. You realize how big of a task that is, and how amazing that we can take something like this, our body, and control the movement that finely. That's an impressive thing, whether you appreciate that or not. So the primary motor cortex is a system in the body that very, very specifically controls certain regions, especially with delicate movements. The third one here I mentioned is a multineuronal motor system. Basically, the difference between the other two is that this is mostly from the brainstem area. This is more non-cognitive types of things. This is more complex than just a basic reflex. By going to the brainstem, it involves a few more bodily systems. It involves a few more different types of inputs, but still it's pretty basic and at like the survival type level. Okay, nothing too cerebral about it. Okay. What do I got here? To control your skeletal muscles in terms of receptors that are there, we have to, in order to get a neuronal input, there's got to be receptors sensing what's going on here. For example, why is it when you tap your patellar tendon down here, whenever you tap that, why does your leg jerk up? Okay, there's got to be some sort of receptor that recognizes that, and that's where we're going with this. We actually have muscle spindles that are monitored by Golgi tendon organs. Let's take a look at the next drawing, show you what I mean. This would be like a bunch of muscle fibers that make up a muscle. Okay, you can see the little A-bands here from all the myosin filaments. Now, within those are some specialized fibers. If you take a look at this, this is called a spindle fiber. Okay, now, I don't have the actual Golgi things listed, but that's okay. This is a spindle fiber, and what you will see is that part of it, like up here and farther down here, can still actually be contractile units. Okay, but within some of these fibers, they are sensitive to stretching, basically. They are mechanoreceptors. Notice the innervation that happens here. These are actually afferent nerves, some of them. They can actually take some sort of, they can be a receptor for what's going on in this area and take it to the central nervous system. So one of the things that can happen is, for example, like how it works with your knee-jerk reflex, realize that the tendon down here actually is part of, it's connected to, functionally, the fibers that make up the muscles here in your thigh, the quadriceps muscles. Okay? As such, if you stretch this tendon, you can actually stretch these fibers that are in your muscles up here. Now, this is not like I'm not talking like an elastic band kind of stretching. This is very small, very minute distances that it stretches. That's all it takes. Okay? So if you can hang your leg rather loosely and tap that for a moment, what you do is you stretch that, and by stretching that, what that means is you stretch, at some point, some spindle fibers that are in the muscle fibers that make up these muscles. When you do that, they stretch mechanically. Maybe there's actual ion channels that respond to mechanical deformation And when they change, sodium comes rushing in. It sends an impulse. It gets fed to the central nervous system. It does a reflex arc, comes straight back, and then an efferent impulse gets sent to the muscle fibers. It spreads throughout, causes a contraction, pulls your leg up. Okay? So these are sort of stretch receptors, these spindle fibers. Okay? Now, well, I forgot what I was going to say now. Couldn't have been that important then, right? Must be. All right, well, anyways. There was something I was going to say. Oh, I know what I was going to say. What's the point of this? Just to entertain ourselves? Why do we have the knee-jerk reflex? I knew it was important. See, that's important. Just to run checks on it, make sure. What's the point in having a reflexive ability to contract the quadriceps muscles? Oh, well, go along with that. Even at a more basic level. Think about it. If I'm standing right here, what you don't realize, we talked about basal cell nuclei a little bit farther and how Parkinson's disease, it shows how some people then lose the ability to control all those extra movements in the body. Okay? We have a lot of extra movements that are constantly being monitored by our body, constantly. One of those is, think about the ability to stand here. Do you have any idea? We are not welded together. Our bones are not fused. We cannot just take out our skeleton, but stick the feet down, spread them out just right, and expect a skeleton to stand there. It's not the way it works. The way that we stand here is through an intricate connection between different muscles contracting at different times, right? So if my knees start to buckle, even a little bit, guess what I stretch? I stretch that quadricep muscle through the same mechanism by which you hit a knee-jerk reflex. Okay? So what does it do? At a very small level, what will my body do? It will recognize that. Those spindle fibers will fire. It'll go to my central nervous system, do a reflex arc, come back, contract this, and pulls my leg right back up. It helps me maintain balance. It's constantly happening all the time in very small degrees. You have reflexes like this everywhere, okay, all over the body. So these are built-in protective things to maintain posture, maintain survival, that kind of stuff. It just is greatly accentuated and exaggerated even whenever you do it and you get a knee-jerk response. If you lock your knees. Yeah, well, that's constriction of blood supply. Well, I guess I don't know what you're asking. If you're asking philosophy in terms of what? Like because of what happens when you lock your knees? Or when you lock your knees, you can pinch blood supply, and as such, you can, like you said, pass out and that kind of thing. I don't think there's, by locking, you're kind of going the other direction. So I don't know of any automatic reflex like this way that you click and then it jerks back. I'm not seeing any of those, but I'm sure there's something at some small level, but I don't know. I don't know. Yeah, okay, now I see what you're saying. I don't know. I'm sure that there is, but as far as for me to trace a pathway, I really don't know. Okay, well, that's surely not something you need to see. You know how that works. Beat it with a hammer till it moves. All right, now, moving on to smooth muscle. I want to spend a little bit of time on smooth muscle, not too long, but realize that the bulk of this unit was about skeletal muscle, and the bulk of the next unit is about cardiac muscle. Those are the very two complicated, neat but complicated types of tissue. With smooth muscle, what you will see is, notice how it's organized. You still have actin and myosin, and you still have multinucleation and that kind of thing. But what is very, very different is it's not striated. And the reason that it's not striated is because all these sarcomeres are not lined up parallel to one another in perfectly organized fashion. Instead, you get like this web of anchored actin, right? You get these anchored actin, and between them, they're sort of loosely connected to these myosin filaments that are already there, and it's sort of like a web of sarcomeric units, but it's not really like a sarcomere, like with skeletal muscle. So what happens is, the difference is, if you can imagine this, Whenever you have a muscle fiber that's got end-to-end sarcomeres, when they contract, what happens to that muscle? It shortens, right? When you have a web like this that wraps around in all directions, and every part of it can contract and pull, guess what it's doing? It's going to pull towards the center. The whole thing is going to come in on itself when it contracts, right? Does that make sense? It's not going to just shorten in one length because it's all wrapped around with this whole web. So when every part of those things come together, the whole thing is going to come together. And this starts to make sense as to why this would be like in our stomach and in our intestine and all that kind of stuff to help pull all this kind of stuff together, not just, it wouldn't be much good if our intestine just went like this all the time. Okay? It wouldn't be very helpful. All right, so it's unstrided, got actin and myosin. Again, you've got the same situation with troponin, tropomyosin, all that's the same. Actin and myosin still working the same way. So we've still got calcium ions that are involved with the bonding sites and all of that. The difference is the mechanism by which that calcium gets released is a little bit different. There's not a lot of that sarcoplasmic reticulum that's stored all of it up like in muscle cells. It's just floating around in the ECF, in the extracellular fluid, and it just gets brought in. Because what you got to realize too is that this is much slower contractions, much less frequent types of contractions, much less forceful types of contractions than skeletal muscle. So everything's sort of laid back when it comes to smooth muscle. Okay? But the good thing is we don't have to worry about thinking about it, and we don't have to control it. It does it itself. Okay. I think that pretty much takes care of that screen for what you need to know. Now, we have what's called multi-unit and single-unit smooth muscle. And basically it goes like with motor units. When I talked about motor units, I said a motor unit was the neuron, the feed that was coming in, and then all the branches, all the fibers that it divvied off and came in contact with. That was all a functional motor unit. Well, smooth muscle can be multi-unit or single unit. And if it's multi-unit, that means it's going to be produced by a nerve. It's going to have a feed coming in, and it's going to branch off, and it's going to touch different areas of cells on that smooth muscle tissue. Okay? It's neurogenic. In other words, the impulse, the contraction itself is initiated by the nervous system. You'll find that that's different when we get to single unit smooth muscle. But here, it's nerve produced, that feeds it, it's neurogenic, it sort of branches off and goes to different areas on that tissue. It is involuntary, it's controlled by the autonomic nervous system. Okay? It's got properties partway between skeletal and single unit smooth muscle, but that doesn't help you until I talk about single unit smooth muscle. So we'll get to there in a second. Where you're going to find this, blood vessels, tubes like in the lungs, that kind of stuff, the ciliary muscle up here in your eye, the iris, the base of your hair follicles. These are all a little more complicated. They don't seem like it, but they're actually a little more complicated in terms of recruitment and the different types of muscles involved. So that would be multi-unit smooth muscle. Okay, now, single-unit smooth muscle, they are unique because they form what are called functional syncytia. Okay, word of the day. Functional syncytia are syncytia, one word, are basically a big web of cells that are stuck together with gap junctions between all of them. Think back to gap junctions, right? They're little holes that they were anchored together, but they were little portals which they could share materials with that goes through from cell to cell. Okay, so we've got a group of interconnected cells, and what's interesting about functional syncytia and single unit smooth muscle is that they are one unit. They act entirely as one unit. They are not controlled like multi-unit from other feeds, and it is not neurogenic. It is actually myogenic. The nerve, the potential, the action potential that leads to contraction is generated within that muscle itself. Okay? Like, for example, you'll see that cardiac can do this, right? You can pull out a heart from a person's body and let it sit there, not hook to any nerves, and it'll beat. It'll still beat. Now, when an action potential develops in one cell, when you have a syncytia, just to know in your head what we're talking about here, it'll be this. Okay? An action potential is developing in one cell. This would be like a single unit smooth muscle. In other words, it's not got various feeds in it. It's localized all by itself. And when an action potential starts, like in a pacemaker cell, I'll discuss that when we get into cardiac, some cells have the ability to control the pace of everything else that's going to be going on. I'll explain to you why when we get there. But they can spread through those gap junctions the ions that could then stimulate the plasma membrane of all the adjacent cells and instantly they function as one entire single unit, not as multiple feeds. Okay? That would be a syncytia, what you just saw, a whole bunch of cells. No, we'll get there in chapter 20. We'll talk about that. Wait, that's my teaser. Just wait. Chapter 20 is very, very interesting. It's the whole reproduction and physiology associated therewith. Really amazing stuff. Single unit smooth muscle is myogenic. It's initiated by the muscle itself. Okay, it's self-excitable, basically. It has the ability to have pacemaker cells that can control and all of a sudden start it. It's sort of like, I don't want to go into explaining it now, but it's sort of like a swing. That when you start swinging, you can move your legs. And before long, you get higher and higher and higher. And if that was actually like graded potentials, before long, you swing up high enough to reach threshold. And then it sort of just swings back down, and then it builds momentum, and it goes back up again, and it reaches threshold again. You'll see that these cells can do that, and it's really amazing that they can do this. Okay, that's what I'm talking about here. Automatic shifts in ion concentrations. They can just shift and keep shifting, gain momentum, and eventually reach threshold when they do this. And I'll explain all of that a little bit later. Okay, so it's nerve independent. Now, that doesn't mean it can't be affected by nerves. As you will see with cardiac tissue, when we get into the heart, that the pacemaker cells of the heart are independent. They function on their own. The heart can stand alone independently from the nervous system. However, in order for you to survive in your environment, you've got a lot of feeds that go in. You may get a fight or flight response where you have to just necessarily increase for that quick moment, increase the amount, your heart rate, increase your respiration rate. You may have high blood pressure, all that kind of stuff. That there are nerve feeds that feed into the heart that override. Whenever they're stimulated, they will override the pacemakers that are present there. Okay, so you get an interplay of the two, but realize that it's still possible for that muscle contraction to happen independent of the nerve supply. All right, let's see what we got here. How many more of these do I got? We can finish this. I've already talked to, the biggest difference here is with single unit smooth muscle, you can get gradations of contraction. You can get varying strengths of contraction. And it has to do with the way that they are designed. They're not in strict parallel lining, so they're sort of all spread out and just grabbed on. Realize that the optimal length for smooth muscle is different than for skeletal muscle, because already as a resting potential, as a resting situation, smooth muscle, especially single unit smooth muscle, is closer together. The actin overlaps the myosin more, so that when you stretch it, it actually is not already in its optimal length. By stretching it, you put it in its optimal length, which is good, because that's how your stomach works. You fill it up, you stretch it, and when it stretches, it reaches optimal length, so that when it contracts, it actually gets a more forceful contraction. Now, you can overextend, and when you do that, you create tension. Up until that point, you've not really created tension. All you've done is created optimum length. Once you go past optimum length is when you start creating tension, and that's when discomfort and all that kind of stuff sets in. Okay? You can read through this on your own. I think that there's not much there other than what I just said. So, I talked about this just now too, so you can look at that. Cardiac muscles. What I'm going to end on here. We're going to focus on that in our next lectures. It's different. It has properties of both smooth and skeletal muscle. It's highly organized. It's striated. So like skeletal muscle, it's highly organized. It's striated. It's got a length-tension relationship. You can only go so far. You'll see that there's something called the Frank-Sterling law you'll read about. As you stretch the heart, it sort of has an elastic rebound, and it comes back and it pumps out. So the more it fills, the more it pumps out, kind of thing. Tons of mitochondria. It's got T-tubules, like we had in muscle cells, the things that come down in and take that thing down to where the sarcoplasmic reticulum is to get rid of all that calcium. It's got those. However, like smooth muscle, it can self-generate its own action potentials, which is unlike skeletal muscle. It has a branching network. It's kind of like how smooth muscle just veers off all over the place. This one's a little more organized, but it actually just branches. It's like a basket, sort of like basket weaving. So take a look at that table that I have indicated there. That's it.